Right, I'm sure a few more people will trickle in, but they'll just miss my exciting introduction. There we go. Too many excitings in this business and with the name of the company like that. Welcome to the next exciting seminar. I'm really pleased to welcome Professor Dr. Thorben Cordes. Thorben and I were just talking before we got started, nearly overlapped physically in Achilles Caponidis lab in Oxford way back when. Thorben is of course now a full professor of physical and synthetic biology in the Faculty of Biology at LMU Munich. Prior to that, he held assistant and associate professor positions in Groningen, before which he was a postdoc very briefly. I think clearly got some great papers and got his job while in Oxford for really only about a year and a half, I think it was in the end, wasn't it, Thorben? And then prior to that, yeah, and then prior to that, it was a postdoc with Philip Tinnerfield in LMU Munich, and PhDs at in a similar institution, although with a different supervisors. So Thorben, I'm really excited to present him to you guys. The work that he's done in the FRET field across many areas has been really transformational. Most recently in the space of looking at the precision and accuracy of FRET efficiencies for accurate distances in protein systems. That's been really important. I'm really looking forward to telling us a bit about that. And of course, one of the big challenges facing people before they do the single molecule FRET experiment is actually generating the labeled material. And this is a big, it's a pinch point, a bottleneck for a lot of people in this process. And Thorben's recently developed a really excellent piece of software that helps identify the best sites for dye labeling. And so I'm really excited to hear both of these things. If you're keen to get more information more generally about single molecule FRET, FCS, exciting instruments and other things, then you can click on or take the QR code there. Then you can sign up for these seminars, and you can also sign up for our Exciting Times newsletter and other bits and bobs. But other than that, I'm super excited. Thorben has said that he's very happy to take questions during the seminar. So if you want to just type stuff in the chat, Ben and I will keep an eye on that and interrupt Thorben as appropriate. So do feel free just to interject as we go, and we'll take it from there. Otherwise, thank you ever so much, everyone, for coming, and I'll hand over to Thorben. Ben, if we can let Thorben share his screen, and we'll take it from there.
So I'll start or taking over the screen. So it says if I click here, then I will prohibit that you share the screen. So I just did that. I hope you don't mind. Tim, thanks a lot for having me here and letting me present the most recent work, some things that are in preprint state. And so I'm grateful for people to come at the quite diverse times around the globe. So my name, as Tim introduced me, I'm Thorben Cordes. I'm from the LMU Munich. And I'd like to introduce myself more from a maybe starting biological viewpoint. So I'm a physical chemist who in the end got into biophysics over the techniques. And I think the reason that I'm bringing up the slide that you see here, which is related to a multi-subunit protein, is merely to show you and make clear why single molecule FRET is an important technique and why we should bother about temporal spatial resolution, reproducibility, matches with structures. So my lab is interested in understanding active import and export processes. So in the end, we bother, so let me just get the laser pointer, we bother how these orange molecules here get across the membrane. And the processes can be from outside to inside or vice versa. And in our case, they're driven by ATP hydrolysis. So this has some relevance, which I think all of us have relevance in their biochemical systems. But I think the complexity that I would like to highlight here in the beginning is such that the transport process, in our case, to work requires really a complex interplay of multiple domains. So what you see first in our case, or what is believed, is that there's an equilibrium binding step of the ligand. You see you go from an open to a closed state. Then somewhat this closed state or the open state or something in between has to interact with a transmembrane embedded part that we call TMDs here, that are going from an inward to outward facing state that is facilitated here by cytosolic ATP binding. And then well-timed hydrolysis is bringing the substrate into the cell, in this case against the substrate gradient. And the model that I'm showing you here is relatively deterministic. People had crystal structures, they looked at the crystal structures, interpreted them in a functional sense, combined it with biochemical data. However, there is no direct evidence that the sequence of events really occurs as we see it here. And this is the case for many systems, many protein systems, macromolecules. So we'd like to collect this evidence. We would like to observe the molecules in action, in movement, and use this progress then later for maybe drug development or others. So each of us will have their own motivation. And the questions that I would say we solved and contributed to, so there are a few people also from my old lab here in Groningen and new people from Munich, were using the idea that, of course, since we cannot capture the three-dimensional structure of a protein, let's assume this substrate binding domain or protein that you see here in the open form. There's a secondary structure representation here where you have one lobe, so that is the left right part of the protein here in gray. The left part here shows the other domain. These are rigid, they are linked by a two-segment beta sheet. This beta sheet allows the conformational change when the ligand binds, and you see in overlay representation the closed state here in orange. And it is quite clear that the distance between two strategically chosen sites differ in this case by about 20%. So if I play this up and down, then approximately 20% change, and that crystallography will be able to capture this. You see it in the structures. However, it is not dynamic. And so people, including us and maybe many of you, use first a resonance energy transfer. So this non-radiative transfer of excitation energy from a green to a red dye or distinct colors as a probe, as a molecular ruler to look at the relevant reaction coordinate. And using this approach, using distance changes in the form of FRET efficiency, so here intensity-based, by looking at the intensity of the red chromophore after excitation of the green chromophore and normalizing it to the fluorescence intensity of both. So long distance would be low efficiency, everything stays at the green chromophore, or 1, everything goes to the red chromophore, will tell us something about the state of this chosen reaction coordinate. So if we monitor these two lobes here, or the inward and outward facing state of the transporter, or here the dimerization of these NVDs. So our lab has contributed to a number of questions related to these transporters, binding mechanisms, what drives conformational changes, what are rate limiting steps. But this more as a motivation of why single molecule FRET, I think, is an interesting technique, because we can combine high spatial and temporal resolution in order to elucidate these types of mechanisms. And what I'd like to show in the first part is where we stand within the community in terms of spatial and temporal resolution, and later how we can also design assays in a more systematic fashion. So here you see two, I think, landmark papers or overview articles that Eitan wrote over the past years. I've also seen him today, which illustrate the approach. So if you're not so familiar with single molecule FRET, then the approach we're taking in many cases relates to an analysis of X-ray structures, making predictions about short and long distances, maybe first in a relative sense, then often using confocal microscopy, where you use the tiny excitation volume of the confocal microscope to obtain information on the state of the molecule diffusing through. So is it in the open state with the long distance, the low energy transfer efficiency, or is it the short distance and high energy transfer efficiency? Since we're doing single molecule detection, we're suppressing background by this confocal detection scheme a lot. We are collecting these informations molecule by molecule, and we can make histograms. So here on the x-axis, you see the FRET efficiency information on open or closed state, and on the y-axis, you see the frequency with that those events occur. The temporal information can come from multiple different types of analysis. One would be to tether the molecules on the surface and then find their location. So here's a 10 by 10 micrometer scan. Go back with the laser to a certain location and analyze the fluorescence trajectories as a function of time, where again, acceptor fluorescence here is red, donor fluorescence is green, and A divided by D plus A gives this black curve where you can see the steps between the open and the closed state, and with that obtain direct information on the biochemical equilibrium as it occurs. So there is no trigger really necessary for this to work. A few years ago, the field, and this started, I would say, already more than 10 years ago, into a more quantitative direction. So here's a paper initiated by Thorsten Hugel, Klaus Seidel, Jens Michaelis, and Tim Craggs, where the question was asked, how absolute can you measure interprobe distances? Because one thing to realize is that in proteins, we're not measuring distances between residues or in this case, nucleobases of DNA, but we're measuring interprobe distances that are somewhat related to the accessible volume of the dyes that can be sampled related to the biomacromolecule. And on the other hand, how precise, so how reproducible and accurate can these results be? So there were many things that were questioned also by other people from more a structural background, crystallography, NMR, cryo-EM, maybe in later years, how reproducible and reliable is accurate FRET. And so there was an initiation of this static double-stranded DNA structure, which was analyzed by multiple labs around the globe with, I would say, a number of different techniques. You will see our most recent comparison study was a bit more restricted in that sense. So the samples were sent around, and I think an important contribution of this paper was also to define a very clear procedure with that the data had to be corrected in order to arrive at accurate FRET efficiencies. You remember that what we measure, at least in the way that I presented it here, are intensities of donor and acceptor fluorophores, but of course these contain a number of setup dependent parameters. On one hand, background, which we subtract here often in the first case, spectral crosstalk, so the leakage of the donor dye into the acceptor channel, but at the same time, the direct excitation of the acceptor dye by the green laser. And secondly, detection efficiency differences between the red and the green detection channel if we use these fluorophores, and also quantum yield differences. After these corrections, you arrive at an accurate FRET efficiency, which by the Förster formula can be converted into an interprobe distance. So here you have the accurate FRET efficiency. Here you have the Förster radius of your dye pair, which denominates the distance, interprobe distance between the two, where you have 50% of this energy transfer efficiency. And there, of course, the spectral overlap, the orientation, and the donor quantum yields play the major role, including the refractive index between the two. That gives us, in the end, the distances. There are a lot of corrections required, and I think it was really a milestone that we have now a reproducible method that, as you will see, as an outcome of this first DNA paper, gave really reliable results between the different labs. Just as a definition here, what are precision and accuracy? Because these are the terms used. So I acknowledge my friend Jan Lipfert here, who gave me this elegant picture of his showing if you're aiming at the head of Jan Lipfert with these snowballs and you do this precise and accurately, then you see where you would aim. If you're precise but not accurate, then you're reproducibly throwing at the same location with a little spread. However, you're not really hitting Jan Lipfert's head. If you're accurate but not precise, then you see you have a large scatter, but in general, you would hit him. So the center of these points would correspond well. And neither precise nor accurate would be next to him, and also a big scatter. So that means that precision somewhat is reproducibility and accuracy that match with the goal or the ground truth of your experiment. And if you looked at the results from this Nature Methods publication from Hellenkamp et al., then in essence, you had about 2 angstrom precision, 5 angstrom accuracy, so reproducibility 2, and match with the structures 5 angstrom accuracy. And this was a pretty good starting point, I think a good demonstration, because it shows that FRET as a reliable technique can really start providing distance-constrained structural modeling as a tool in integrative structural biology. When I started my lab in Munich, or when I moved the group, we wondered, how does this translate to proteins? So here is a snapshot of one of the favorite players in the lab, the maltose binding protein, with the parameters obtained from, or input provided as an input to the accessible volume calculations from Klaus Seidel are provided. So you have a linker width, linker length, and the fluorophore represented as a bit of an ellipsoid, which in the end gives you here, I think, a good demonstration of the differences between the residue distance or the interresidue distance of 2 cysteines that we have selected in this mutant, and the resulting measured distance between the 2 probes. This can differ up to 15-20 angstrom, so I think it is quite considerable to be taken into account. But of course, this is not specific to proteins. Why should we even bother and think that proteins behave differently? Well, at first, this was a training project for a physicist, Christian here on the left, and Rebecca helped him with making the initial mutants. And the additional hurdles that we saw in the beginning were that proteins are, in contrast to DNA, rather dynamic, so they can have motion on different timescales. We wondered how would that impact the accuracy and precision. On the other hand, conditions, salt, pH, storage time might influence the results we can get. But what we also realized at one point, that we use stochastic labeling. In the case of double-stranded DNA, and you buy fluorescently labeled oligonucleotides, you have the probes attached really specifically to one location, so you have site-specific attachment. But in many cases, I think the predominant method in proteins is to engineer 2 cysteines, label them with melamides, and use a stochastic mix. However, if you look, for instance, at the accessible volume clouds using the FPS software and compare Alexa 546 and 647 in their locations, right and left, you can see that there's really a difference in the size of this accessible volume cloud, particularly here of the Alexa 546 on the right side. So we really reasoned that there will be many different problems that could impact the results for proteins. In the end, I think this is the summary of why we thought this was interesting. And of course, I have to mention that we got a competent team on board. Klaus and Don and also Anders, Bart from the FRET community joined us on this endeavor over a few years, really testing the, let's say, questions on how well can we measure FRET efficiencies accurately in proteins. So this was published just this year, and it was testing the FRET ruler with this maltose binding protein. We started with these 3 mutational pairs that you see. So the first one is a front mutant that has these 2 labeling sites where, from the structural transition from apo open to holo closed, you see there's a shortening of the distance. And this is reflected in a transition of the FRET efficiency from moderate to slightly higher. If you check the affinity of the protein, it's around a micromolar KD, and this is well reflected in the 50-50 mix approximately at this concentration here. We can do the same at the back side of the protein in the hinge region or the balancing interface, depending on the paper you look at, where you go from short distance to long distance. So here, high FRET efficiency to short FRET efficiency, again, affinity preserved, or go to a location where you have no net change of the interprobe distance. These samples, we didn't tell anyone what they were. We sent them around to really a large number of participating labs and obtained, as you can see here, the results as displayed, where I think we have relatively nice reproducibility. You have low mean values and standard deviations, which actually turned out to be quite similar to double-stranded DNA. I should say that the maltose binding protein is a protein with relatively slow conformational dynamics. So there's of course no question that if you have a state that persists for about 100 milliseconds or even longer, then the distance, the mean FRET efficiency that you determine in one state would be directly related to the interprobe distance in that state. However, the question is what happens when you have a protein that has much faster dynamics, let's say faster than 10 milliseconds. And here we had a second protein system where we got help from the lab of Don Lamb and also Sattler from the TU Munich that provided an RNA binding protein, U2AF65, which has this very flexible linker and also two domains, which upon RNA binding transit into a more stable hollow conformation where the linker, despite its flexible nature, allows the two domains RRM2 and 1 to be fixed. And we did the same. So we looked at the FRET efficiency distributions of the APO, so ligand-free form and RNA-bound form. So you can see you go here from a high FRET ensemble that is relatively broad. So there are likely many different states underlying this distribution, transiting into a low FRET or intermediate FRET form that is stabilized with a bit of the high FRET state persisting in this state. And also here, we asked people, please provide your mean FRET efficiencies, fit them with an appropriate state model. So everyone, you see it's a smaller set of people that responded to the second round of analysis, where again, I think we get quite nice reproducibility of the data. And also here, we are in the range of just a few angstrom, slightly worse than in the MAL-E case for the mean FRET efficiencies in this case. And then, of course, the additional information we needed from the labs was based on the idea that we needed to know, would you characterize this sample as something that is dynamic or static? Because if the sample is dynamic, or at least in one of the states shows dynamics, sub-millisecond dynamics, then likely a conversion from mean FRET efficiencies into an interprobe distance is physically not meaningful. So we used two techniques, or most of the labs reported based on two techniques: burst variance analysis from the Kapanidis group, and also e-tau plots that I believe were introduced from Klaus Seidel's lab. And we asked people, please look at the state of the different proteins, the biochemical conditions, and report back, what does your technique say? And what we found, I think with an overarching majority, I think there was only one lab that did not agree that MAL-E, the maltose binding protein, is not static. So we believe this is a relatively static protein, in contrast to the U2AF65 in the APO state, where we saw really a huge deviation of the burst variance. So basically the fluctuations of the FRET efficiency or the donor and acceptor signal during the transit of the molecules through the confocal volume. We did that also with statistics, looking what are the expected values, what is predicted based on the structures, taking the extremes, MAL-E open, MAL-E closed, and also doing modeling for the U2AF65. And this agreed reasonably well. You see there's, based on the predictions and also the measurements, a bit of more deviation than with the pure measurement of these mean FRET efficiencies. However, I think we had an overarching agreement on the dynamic character of the RNA binding protein and the non-dynamic character of the maltose binding protein on timescales below 10 milliseconds. Similarly, we got results from people that did e-tau analysis, so where you look at the dependence of FRET efficiency versus lifetime. So those labs that used pi or nanosecond RLEX in contrast to the slow microsecond RLEX implementation had, I think, very similar results and interpretations. So I think what we could also show in the paper that is relatively important to support our claims that we can really contribute towards structural biology quantitatively is that we can really reliably detect these types of dynamics based on different methods. The quantification of relaxation rates and so on was not done by many labs, so there we don't have statistics yet. So this is still something where the community, and this may actually be a call maybe to a lab that is interested to go into this direction. I think this is probably one of the next steps that has to be taken to look at how reliable can labs determine, based on some protein systems or DNA hairpin, etc., kinetics, relaxation kinetics, but also really rate constants. Okay, so what are the general limitations for temporal resolution? We did a few tests in the beginning of the project. So if you ask yourself for the MAL-E protein, if you look at immobilized molecules, as I outlined in the beginning, how well do, for instance, the immobilized FRET efficiency traces match with the mean values we get from different labs? We had a subset. I think this didn't even end up in the paper in the end. However, you see here time, here accurate FRET efficiency, and you see the trajectory of a single MAL-E molecule approximately at KD concentration, so the protein samples between the open and the closed state. And what we did, we mapped here the mean FRET efficiencies from, I think, a subset of 5 to 6 labs in apo and hollow state. And again, we have a relatively nice agreement, which I think shows you that we can even measure these distances, the accurate distances, at a speed on the order of milliseconds. And I think it's clear from contributions of Gilad Haran, but also Eitan Lerner and others, that you can even push this further. There's PDA and other techniques, but one I'd also like to highlight and mention that Eitan Lerner's lab published, where you can do a state analysis on the timescale lower than the burst duration, where you can still extract these transition rates based on hidden Markov modeling. And if you're not interested in rates related to FRET efficiency states, then of course you can push it further if you use FCS, which I think Ben Schuler, Don Lamb, but also Klaus Seidel and others have used and introduced, where you can really go down even to the sub-microsecond stage. So I think that shows you that FRET is really a unique tool from a viewpoint of quantitative analysis of structures, from a viewpoint of one reaction coordinate currently, but at very high temporal resolution, which I think is really one of the unique features of this technique. Okay, so if we know now what the FRET efficiency, the mean FRET efficiency represents that we measured, we analyzed now the MAL-E FRET efficiencies, converted them into distances, made structural predictions based on accessible volume calculations. So Klaus's group did the major effort here, and we of course compared the computed distances versus the experimental distances, or vice versa in this plot, with a comparison of the two, seeing that in some cases we could use the accessible volume, so the accessible volume of the dye with unrestricted motion, but in other cases, particularly when we could demonstrate later with anisotropy experiments, in some cases the accessible contact volume was a better representation for dyes that would actually stick to the surface. So this particular variant here, the K29C and S352, as you can see here in the anisotropy decays, actually had a larger component where the dyes would stick, and this was particularly problematic for this 352 position in combination with the acceptor dye. And here I think you see that it is relevant, particularly if you are trying to obtain information in relation to structures that you carefully check the dye properties on the distinct positions. We've tested a number of dye combinations, also tried maybe to provide some, I would say some guideline on suitable dye combinations. However, I think this is also still a topic where people could really dive into and find out, is there a dye combination or a number of dye combinations that is most attractive for FRET studies and gives really the most reliable results. And of course, I won't make too many statements here. I don't want to give complete advertisement to the dye suppliers. Another side note that I'd like to give in the end, so we also analyzed or characterized the comparison of FRET versus EPR with the group of Gregor Hagelücken, where the question was, how do the conditions of freezing and labeling proteins with spin labels versus our approach of using intensity measurements and conversion of these FRET efficiencies into distances, how would they differ? And I think one of the major implications that we expected were based on the size of the spin labels, and in that case also the resulting accessible volume clouds, also the predictions of EPR distance distributions are based on Rotterdam libraries. So this is something that has only been very recently introduced for FRET labels, but it is also available by now. And what you can appreciate, I think, in this image here, we used the maltose binding protein, but also 3 other protein systems to do a benchmark. So here you see, for instance, the EPR distance distributions, black thick lines. So this is experimental data from Peldor or Deer versus the mean FRET efficiencies that we obtain. You see there are quite a number of differences in the absolute values, which you might explain by differences also in real measurements, since we deal with different spacing of the dyes because of the accessible volume clouds. But overall, we found a relatively good agreement with larger errors between FRET and Peldor. So if you look here at the black dots, this is the experimental deviation between FRET and Peldor in the experiment over, in this case, 15 experiments. And you see the fluctuations are in the most cases on the order of 10 angstrom or so. Some deviate larger, but I think we have a relatively good qualitative match and a nice feeling after this paper also that the techniques provide complementary yet consistent results in that sense. Okay, at that point, I'd like to look a bit in the future and also motivate how we came to the development of this in silico prediction tool that I'm going to introduce. And one reason for thinking about an in silico prediction tool came from the fact that I've said that we can use FRET for a number of different interesting studies. I probably don't need to tell you that we can use it for monitoring of interactions, conformational changes, and many others. However, what happens if you would like to visualize both at the same time? Then, well, one possible solution is to go to 3-color FRET, 4-color FRET, or approaches that require an additional order of magnitude of label complexity, because it becomes really very difficult to attach the label in this combination. Often the yields for 2-color labeling is already limited. Data analysis becomes more difficult. Chromatic aberrations play a bigger role. So what we've been thinking about is a simplification of this approach of using FRET based on either environmentally sensitive dyes. So here is a trial of a sensor for maltose. So we made this maltose binding protein into a 1-color sensor based on environmentally sensitive dyes, or also using contact quenching between 2 fluorophores. And these approaches, at least the upper one here, has also been shown by Takship Ha more than 10 years ago for enzyme studies to look at conformational dynamics. And the main idea in the lab here was to try to visualize at the same time correlated motion or subunit interactions plus conformational changes. And this has been successful to some extent. So for the maltose binding protein, we made a number of single site cysteine mutants, a number of double labeled site mutants for quenching, or in this case, environmentally sensitive dyes. However, the prediction possibility of this was, I want to say, very poor. And also when we wrote a review, so the credit here again goes to Ethan Lerner and Stephen McGinnis and Evelyn Plutz, who started this overview on PyPhi. So this is a technique where, for instance, the change in accessible volume here of a green cyanine chromophore changes the isomerization rate, and with that, the brightness of the dye. In a nutshell, for instance, when you have binding of a protein molecule in the vicinity of this dye. However, if we just used accessible volume calculations for predicting when we want to see Pi-Phi effects in proteins based on conformational changes, we had pretty much no success with this strategy. So the question for us was, how can we select suitable residues in proteins systematically? And at that stage, we realized we actually have to really take a step back before we think about the assay, because this is what Christian also did during his PhD, and this was work with Jan Lipfert, where the first step in the process actually has to be not selecting a residue that is only suitable for the biochemical assay. The assay here would be to detect the arrival of this DNA binding protein, but to select a labeling site that doesn't mess with the protein, the secondary structure, its function, or in, let's say, a bit of a less worse scenario, maybe that the fluorophore interacts at a specific site. Maybe the charges are completely bad, so that there's sticking of the dye. We've seen in FRET this would correspond to higher, to lower accuracy with relation to the structural model when you do the measurements, or that you have weird quantum yield of the dye because maybe it is really restricted or quenched by an amino acid such as tryptophan. And we have a web server. The web server is here, and I have to make a bit of an embarrassing claimer at the beginning. At the moment, the server has a problem that we were not able to solve until today. So I've worked with 2 students over the weekend trying to solve this, but unfortunately, I can give a short demonstration of the capabilities of the server, but we have a communication problem with one server that calculates an important parameter. And this important parameter that we consider before laying out the assay is the conservation score. So if we're thinking of how to label a protein, then the sites we want to select, and this is what most of us do, and what we've been doing, I would say even rather successfully in many cases, is to look for solvent-exposed residues because we want to attach a dye. We also don't want to touch a residue that is really relevant for the protein. So a means of doing or assessing this is to look at the conservation score to see what is exchanged in other protein homologs in other organisms, to avoid to use residues that have relevance in secondary structure, and also see what is the nature of the amino acid that we're going to replace towards the labeling site for the cysteine. And then we took a base approach. So we said, okay, we calculate the odds ratio for these parameters to occur in a residue that we know is labeled. So your question has to be, where did you get this information from? And this information we obtained from your publications, from some of you at least. We've looked at papers, or Christian looked at papers and saw what residues in proteins could successfully be labeled and used for FRET and other assays. We identified about 400 of those residues and calculated the probability distributions that these residues that we found could be labeled versus the probability distribution of these values on the left over all residues in the protein. Yeah, and this we took as a first indicator to predict if a residue is suitable for labeling. The database contains a number of different proteins. There's an Excel file that we will provide with the paper in the end. You see here some statistics on the database, how many residues per protein we had, what type of assay has been done in the literature, if it's a soluble or a membrane protein, and what kind of mutation has been done. And here are some key statistical parameters. And what we did is we compared, of course, also this data set of selected structures or proteins to a random selection of proteins from the PDB. So that's this PDB select from now 6 years ago. And in the end, also looked, this is what you see here, for the diversity of labels. So you can see here the most popular labels are these cyanine-based Alexa fluorophores 555 and 647. Not that they're necessarily the best, but this is what the statistics, at least in the database, say. Okay, what we do then is we provide as an input only sequence and structural information. So what you need in order to do anything I'm going to show you is you need a PDB structure in essence. This PDB structure is put into our Python based analysis. So there's also a Python-based code that is independent of the web server that I'm going to show you that calculates the label score. The label score is in essence the geometrical means of these different parameter scores that we have here, which represent the properties of labeled residues from the database in comparison to the non-labeled residues. Here, the labeled score, I'll show that on the next slide, is represented in comparison to the different parameter scores. So you can see what it results in. And a redder shade of this label score indicates that these positions are the ones that our algorithm predicts to mutate into cysteines and later use as labeling sites. And if you compare the label score of the residues that we have in the database, which were labeled, versus the random selection in gray from the PDB select, then I think you see that there is a large enrichment in scores above 1, which says that the database that we have selected contains, in combination with these parameters, really information on the suitability of this position for labeling. If we now take this, so here again, the maltose binding protein with the label score, and we look at label efficiency. We thought, is there a correlation between label efficiency and the label score? You see, this is not really a correlation. However, we think this is not expected since we are in a biased experiment here. So we haven't randomly selected these single site cysteine mutants, but we basically took what we had in the fridge, testing the label efficiency. And that means that we were likely biased towards high label scores, and thus there might still be a correlation, but we haven't tested this really. Also, the label efficiency proved to be independent of the dye that we used for single site labeling here, which also tells you this is, I would say, good and bad. Well, maybe the label sites are all really suitable. The dyes were all similarly reactive, maybe the site 3 not in this case. However, also our approach doesn't take into account the properties of the labels or even the interactions of the site with the labels itself. What you can, however, do to exclude, for instance, photophysical artifacts or minimize them is to give also amino acids and exclude labels from this score, so suppress their score. If you don't want tryptophanes or methionines in close proximity, there's an option to basically take them out, because these are the ones that give you a photophysical reduction in stability. I think there was a very nice JAX paper from a Chinese group recently, which demonstrated that methionines and also electron-rich melamide groups are likely the cause for lowered photostability of protein-linked fluorophores. So what we did then is we say we have a set of label sites that we consider suitable based on the parameters that we have provided as an input. Let's test it. So what we did is we used it in our favorite assay, and it is not a big surprise that this is FRET, where we tried now to implement the accessible volume calculations in a slightly more or slightly faster way, so computationally more efficient. So we used a segmented sector model, which uses basically looks how much of a sphere is cut by the protein, just to make the calculation of the accessible volume faster, and then defined a second score that really is independent of this label score, in a sense that it now considers the properties of the labels, of the label residues based on their LS score. So there is a multiplication, and we are trying now, based on the position that we have selected for these two residues, to get the value of FRET efficiency as close as possible to 0.5, so to arrive at a distance, an interprobe distance close to the first radius. And so we get a high score if the label score values of these residues are good, and if the predicted FRET efficiency is close to 0.5. Of course, what is also quite relevant is to compare two different structures. So if you have an apo and a holo state, here is a phosphate binding protein that we chose as a model system, which we had not studied before. So we take here the label score values of both residues in the two states and maximize the distance in the predicted, the difference between the predicted FRET efficiency in both states. And this we define as the FRET score, and this is the FRET difference score. And this works pretty well. So we have used it for really a number of different proteins in the lab, not only the one that we put into this paper. So here are 4 different mutants integrated in the paper. It's available on bioRxiv, which shows that I think there's a quite reliable prediction of FRET assays possible based on the analysis. And we've also seen if there's a correlation between these FRET and FRET difference scores with experimental values. So in black and white, basically, you see here the correlation with the all distance measurements from our comparison study that was published at the beginning of this year, and in red, some new experiments that we provided, and here on the right side, the correlation with the FRET difference score. So I think you can see that there's a huge potential for systematically designing finding the label positions and translating it to an assay. And in the end, we would like to benchmark this now, see to extend it, because I just said that we have problems with one of the servers predicting conservation scores. So we're trying to find alternatives, and also would like to extend this towards PyPhi and quenching. So this is what a new PhD student is currently doing, and hope that at one point in time, we have a large enough database to also do machine learning, because the main reason that we've used this Bayes approach so far is because the data set that we have acquired with 400 positive residues and no negative residues is too little for this. So this is also maybe a call out to you as the community to please provide us with feedback after the server is fixed and you can use it again on the positives, but particularly also the negatives that you have found based on the analysis here, so that we can improve the database. Based on what Ben has shown me before, I will skip this part here, which is photostabilization. I just wanted to advertise one paper on making dye molecules functional. So here we've just published this last year, where we use linker molecules to tether proteins, DNA, live cells, whatever you like, to the same dye molecule and make it either stable for FRET, or you want to make it maybe blinking based on copper chelation in this case. And I'll skip that. You can apply it in FRET, in super resolution microscopy, but I would like to come to an end. Thank the group, because what I would like to transition into is a bit of a live demonstration of the Labelizer on the web page, and show you what it can do. But I would like to close with thanking the current group. So this is a most recent snapshot. Our collaborators, locally, international funding. I've tried to highlight everyone at the specific point. And Tim and Ben, you have to tell me now if we want to do a bit of a question round now, or I should go a bit to the live tutorial.
Let me ask a couple of questions that come through the chat, Thorben, while you set the...
Sorry, I have to switch on my speaker again. Yeah.
Can you hear me now?
Yeah.
Brilliant. Thank you. First, thank you very much indeed for that excellent overview. And why don't I ask a couple of questions that come through the chat while you get the next thing up. And if anyone would like to add any questions to the chat, now's a great time to do that. So we had a question in from Suzanne Zelga, just saying a very important question. I'd like to know if the labeled MAL-E with the additional labels and the cysteines still behaves as the original. Can this be tested?
Yeah, I think this is a very important remark, and I annoyed the group with this very much, because Christian at one point said, okay, we have to get this out now. And we measured the affinity of the MAL-E's in triplicates with all of the variants that are in the paper. So they are, yeah, they behave as wild type. They are all in a range that we cannot distinguish between 0.5 and 1.5 micromolar. yeah, so they behave as the wild type. So this was for me a particularly important aspect, because of course the idea of selecting the positions is also to maintain the function, and this was the only readout in that sense we have. Of course, we haven't tested them for downstream biochemistry, but that we verified.
Great, thank you. Any other questions anyone would like to ask at this point in the chat whilst Thorben's setting up the next part of the... Otherwise, we'll crack on and take any other questions at the end, Thorben. Thank you.
All right, no worries. Okay, so here here you see the limitations that we have. So I guess it was really a bit embarrassing, but at the moment the server is not operational because of these problems with conserve. So I'll try to highlight on our Twitter page whenever this problem is resolved, I hope within the next couple of days. What I can show you is how easy the analysis is with stored and sort of already processed data. So here you find the web page. There are not many buttons to click. There's analysis, there's citers, who are we? So this gives a bit of an overview, a quick description. Also goes back to the papers you should cite if you use our analysis, because we rely on servers and input from other groups. So please acknowledge not only our paper if you use it, but also the additional papers. There are some facts here. As I said, we really would like to obtain feedback from you on the success or non-success of the procedure. Also, if you have, for instance, failed experiments and you would like to analyze your structures in the future with the software, and you can provide negative input, so residues that did not work, maybe with low label score values, this would be extremely relevant for us. Okay, so let's go to analysis. What would we do? So here we could write mal-E. Today is 27/2023. Apo versus Holo. So this is the comparison I can make. So here you can have three analysis modes. You can have a single label analysis. Here we basically provide the label scores of the protein. We give you an output that tells you which residue you can pick and select to convert into a cysteine and then attach a single label. So there's no FRET assay, but you know where to basically modify the protein. You could go to single FRET. So in this case, you enter a PDB code. So here is the 1-OMP. So this is the open structure of the maltose binding protein. If you have multiple chains, then the idea is that you can select only one of them, because the analysis is typically done between only one of the asymmetric units. What I would like to show now is that if we have two structures that are distinct, so the analysis here is between the open and the closed state of this maltose binding protein, which is directly obtained from the PDB. So you just need to know the code, but there is also an option to upload a model that you have generated, or you can also use an AlphaFold model that can be directly uploaded with the additional information. Then you select your favorite fluorophores. You know from the overview in the database that the favorite fluorophores of the community are Alexa 555 and 647. And you also have to be aware that, of course, the first radius which you have here, which you can change, might depend on the local environment. These are cyanine dyes, so the quantum yield and thus first radius can really vary. And you can, in principle, also change this. Then, if you're an advanced user, there is here a window in which you can tune the weight of each of the parameters: conservation score, solvent exposure, cysteine resemblance, and secondary structure. And you can give different weights and different sensitivities, or you can even exclude amino acids. So if you, for instance, want to exclude methionines or tryptophanes within a certain distance, then you can add this as an additional parameter, but I'm not going to let this run through now. And then the only thing you have to do is start analysis. The server hops to this window, and what you would do is you would subscribe to the results. So I put in my email, if anyone is interested to email me afterwards, this is it. So it's cordes@bio.lmu.de. You click subscribe to results. You see this feature still works. And you will retrieve the link that you see here in an email that gives you the information. And I've pre-processed this for the maltose binding protein and the comparison of these two states, as shown here. And you see here on the left the open and the closed structure, so the comparison of the two. And what I'm showing here now is really a long list of results. So what you see here are, on one hand, CSV files with the individual parameters, cysteine resemblance, conservation score, secondary structure, and amino acid identity. Then you get a list of label scores for subsets and the complete data, the complete protein residues. And then here you have the FRET scores, and there are two versions of this. I can't go into much detail. One is long, that contains all residues that are considered against each other, and the refined is using the accessible volume calculations from Klaus's software for the best 300 residues, because this is computationally more expensive. And what you can do then is you can rank the score. In this case, it's the FRET difference score that we've looked at. So we're maximizing the difference between conformational state A on the left and B on the right. And the best residue that the program suggests here is 29,211. So you can see here the 29 residue is one that we have considered in our analysis before, not the 211. I think there is, in this case, if we what we can do now is we can also sort it via residue 1, and we can look at 29,352. So this was the residue that that we have used in the Nature Methods paper. So you see this takes a bit. There's actually quite a lot of residues to scroll through. Just to get an idea in what range this score is. You see this is actually relatively low compared to the highest score. And now you have to think the measurements in our Nature Methods paper were done with the fluorophore pair Alexa 546, Alexa 647. And this has, of course, a much higher Foerster radius. And you see the distance 1, it starts at 70 angstrom and ends at 60 angstrom. So it's really away from the Foerster radius, at least substantially, for both states. So what we can do is, if we do the same analysis now for the Alexa 546 and 647 and do the same. So we sort it via, oh, sorry, I first have to select the correct data set, then sort it via residue 1 and find our 29,352, which is here. So this gets a score of 0.66. And we can look now how well this guy performs in comparison to the overall score, going to the top. And you see here that in this case, it actually ranks as one of the highest ones in the list, because we have a proper match between the Foerster radius and the anticipated differences of the software. So this, at least, I think within this short amount of time we have left, shows you a bit of the capabilities of the software. You can then also click to see where are the labels. So there is a, I think, relatively easy web plugin to visualize it and give you some sense of what you would be doing to the protein. Okay, at this point, I think I tortured you guys sufficiently for today. And I just need to find the button to, here, to stop my my screen from sharing, or at least to ask into the round again if there are further questions.
Brilliant. Thorben, thank you ever so much indeed. That is really great. And I think this is going to be a really important, useful tool for the community as a whole. And making these things available on web servers and accessibility is absolutely key for all of this. Are there any other questions for Thorben? I think we may even be able to unmute and take them live, although it might be easier to put them in the chat, but if you wanted to ask anything, very welcome.
Yeah, if not, then I would say please keep an eye on our Twitter page and have a look when the server is available. I think we would be very happy if soon people would provide feedback on this. I think the service is not useful if no one is using it, so I think we would be very happy to obtain feedback.
I have a question for you, Thorben.
Yes.
How long can it take to receive your results after submitting your request?
I can go back to the results here. So you can read. So the results can actually take minutes to hours, depending on the time that it takes for our server to get the results back from the Conserve server. This is the most heavy workload, and we rely on the other server. We've tried to replace it with variant effect predictors that are much faster. We had a bioinformatics student trying this, but unfortunately, the Conserve server persists to give the best prediction results. So these differences in our database between the labels and the random residue remains the biggest if we take, or the largest if we take the Conserve server as an input. So at the moment, don't be bothered if it takes even overnight to get the results. You will get an email notification once the job has been processed. And if there are really problems that you don't get a result after a day, I think it would be nice if you emailed us directly, and we would, because we can see what jobs are in the queue. And at the moment, I just ask everyone not to submit jobs because we are trying at the moment to fix the interface.
Henry's got his hand up.
Yeah, thanks. Thank you for your nice talk. I just have a short question. We used your tool to analyze polymerase, which is kind of a big protein, and we choose 2 chains in the PDB, and we got scores over 1. So what is exactly this meaning? Is it just so it can be over 1, or was it just an artifact of whatever?
No, the predictor gives you results scattered around 1. So there are scores that range from, so there are some scores that can't be calculated, then it typically gives a 0, and the scores are between 0.5 and 1.9, I think, for the best label score. So this is the expected distribution. If you're a bit uncertain about this, I could send you also, we have this supplementary data set that I couldn't upload to bioRxiv, but I have it that I could also send to people, where I plot and provide the label scores for some of the proteins that we've analyzed in the paper, so that you can also see the distributions yourself, and maybe even have a look if the scores that let's say based on your analysis are reasonable. But also, I'd be very happy to just look myself with you if you would just like my advice, because I've been staring at these distributions for a while now before submitting the paper. So I'd be happy to also help people getting started on this, because we really want it to be used. And I should say also, the idea to make this web server was not mine. This was Christian's idea to say, this is the easiest approach to let everyone use it, provided that the server works functionally, of course.
Okay, thank you very much.
So if you like, just drop me a mail, and I'm happy to provide also input on that.
Yes, sure, I will do that.
Brilliant. Any other questions for Thorben? Otherwise, it remains just for me to say thank you again, Thorben, for a fabulous talk, and the work. I mean, so much work involved there, ultra collaborative stuff, really driving the field forward, and really is a crucial contribution and a key part of the community. So thank you for all your work in this. I'd just like to bring your attention to the next talk, which is actually only in about 3 weeks' time, with Professor Liz Rhodes from UPenn. And she's going to be talking about all things FCS, probing functional mechanisms of complex proteins in a complex milieu. That's going to be all about how to use FCS to uncover complex protein dynamics. Thank you everyone for coming today. Thank you again, Thorben, for your great presentation. And I look forward to seeing you all in a few weeks' time and at various conferences, hopefully.